Now, we did mention briefly about the plant essential nutrients and whether they're mobile or immobile, but we're going to basically mention all the essential nutrients, what they are, and give you a brief idea of how they look in the plant when they are deficient in the plant. And this will wrap up all 4 sections of this talk. So what is in a plant? Maybe this slide should have been in the beginning of the talk, but it's towards the end. And basically, most of a plant is water. 70–80% of a plant is water. And so by weight, when we ship things, it's usually the water that adds a lot of weight to a plant. Organic matter is the carbon that's in the plant, and that's taken there from the process of photosynthesis. The amount of organic matter is also lost through respiration. But only 3–15% of a plant's total weight is actually the minerals, the plant essential nutrients that are in the plant. So it's a very small fraction. But getting those minerals, those nutrients to the plant when the plant needs them is very important and will dictate whether you have a healthy and successfully growing plant. So the plant requirements for these different components: the air will provide the carbon and oxygen through carbon dioxide that goes into the stomates. But the soil will provide the water, which is H2O, hydrogen and oxygen, as well as oxygen. That's why it's important to have oxygen in the rhizosphere, because it is taken up by the roots and it's important for respiration. But then the soil provides essentially all of the nutrients: nitrogen, phosphorus, sulfur, calcium, chlorine, magnesium, potassium, molybdenum, manganese, nickel, copper, zinc, boron, and iron. And as things progress, sometimes some of these nutrients are taken out of the essential plant nutrient column and other ones are considered beneficial. But these ones are considered essential, and up until the late 1980s, nickel was added. So surprisingly, some of these nutrients were added in the past 30 years. So it's interesting to know how these nutrients are taken up and how we get them. Now, in the ornamental and floriculture industries, we deal with many thousands of different types of plant species and cultivars. And it's really impossible to say that all plants have a certain amount of a nutrient in them. With many other commodities like citrus and avocados or peaches, they actually have templates, they have standards for what a citrus leaf should have, and you can follow that. Unfortunately, in the ornamental and the floriculture industry, we don't have those kind of standards. So we have to go with general guidelines. In a dry leaf tissue, this is dry leaf tissue concentration, not the whole plant. Now, when I'm giving these examples, I'm giving you the list of the macronutrients, and macronutrients are those nutrients that are present in the leaf, dried leaf tissue, and when they're reported at the lab, they're reported as a percentage because they're that high in the plant. They make up a percentage of the dry weight. Now, nitrogen is the element that's in highest concentrations, and that ranges from 1 to 6%. Phosphorus is ranging from 0.2 to 0.5%, potassium 1.5 to 4%, and sulfur is 0.15 to 0.5%, calcium about 0.5 to 1.5%, and magnesium is 0.15 to about 0.4%. Again, this can vary depending on the plant type, and it can even depend on the cultivar. And it can make a significant effect on cultivars. I've seen cultivars of plants that actually were suffering from nutrient deficiency symptoms when cultivars of the same species next to them were perfectly healthy. So yes, you can have differences even on a cultivar level. And I have this pneumatic device to help remember what the macronutrients are. And in this case, mine is Nancy Peterson's kite sails calmly and magnificently. And that will give you all the macronutrients that a plant has. They're presented in a percentage. Now, before I mention that all nutrients are taken up into the plant from the soil, sulfur is one exception to that. Nitrogen can also come from the air slightly. But sulfur, there's so much sulfur that comes down in the soil in industrial areas that up until the air has been cleaned up, many industrial areas, sulfur deficiencies never occurred because sulfur came in from the pollution. This is also the case for nitrogen sometimes. Nitrogen, there's so much in air pollution that deposition can be really high in some urban areas. But it still enters the soil and is taken up into the plant. So what about micronutrients? What are the micronutrients? Okay, micronutrients are present in the tissue in very small quantities. And so small, in fact, we present them in lab reports as parts per million. And 1 part per million is 0.0001%. Now, if you were to look at a lab report and try to figure out what these concentrations are, you're going to have to work through all those zeros. And it's very hard to see when you have differences or problems in the plant. So we've presented them as parts per million instead. So it's easier to see when you have differences or ranges. And so an example of this would be iron. Iron, which is one of the micronutrients in highest concentrations usually in a plant, are 50 parts per million. That would be 0.005%. It's a little easier to look at 50 parts per million than 0.005%. So these ranges are really low, but they're very important. Just because they're in the plant in small concentrations doesn't mean their importance isn't any less. A lot of the micronutrients serve their purposes in other enzymes to regulate the other macronutrients. So if they're not present, you can run into a lot of other problems. So the way to remember these micronutrients is this other pneumatic device is fertilizer management cuts back the zone of most clutter of chemicals nicely. I put that in there because a lot of times we're dealing with nutrient runoff in an industry, and so this is one way to kind of insert the nutrient management thing in the nursery industry. But the micronutrients are iron, manganese, copper, boron, zinc, molybdenum, chlorine, and nickel. Iron is usually in the range of 50 to 75 parts per million. Manganese about 10 to 50 parts per million. Copper 3 to 7, very low. Boron 20. Zinc 15 to 50. Molybdenum 0.15 to 0.3 parts per million. Chlorine 50 to 200. And nickel 0.1 to 1. So I don't want you to get the misunderstand this concept here. When I talk about the macronutrients and micronutrient concentrations, I point out these concentrations. But what's also important is not just the concentration, but the concentration of one nutrient to the other. It's the balance of these nutrients within the plant. Sometimes the range may seem adequate for a nutrient, but it may be much higher than another nutrient, causing another nutrient to have deficiency symptoms. An example of that would be magnesium and calcium, for example. If you have a lot of calcium, you may induce a magnesium deficiency. Or if you have a lot of calcium and potassium, you may induce a magnesium deficiency, even though in the tissue analysis, magnesium may look adequate. So it's important to get these concentrations down and get the ratios of one to the other. And what's also important is to keep records. It's not just a snapshot in time, it's a snapshot over time. You need to keep records of different stages of crop development depending on what crop you're looking at and what you're working with. If you're working with plug production, you want to take whole plugs and analyze those. If you're working with more woody crops where they go through vegetative and flowering periods, you're going to want to take samples during the same stage of development in the plant so that you can relate it correctly back to the records from the previous season. So in review, mobile versus immobile nutrients in the plant. We talked about this a little bit before, and that the mobile nutrients, when they're deficient in the plant, will be remobilized from the oldest tissue as shown on the diagram on the left, and then the new growth will stay adequate with sufficient ranges and won't have any deficiency symptoms. In micronutrients, the deficiency symptoms show up on the new growth because the old growth cannot remobilize those nutrients that are stored in those oldest leaves and remobilize it adequately to meet the needs of the new growth. And again, the mobile nutrients are nitrogen, phosphorus, potassium, magnesium. I have molybdenum and nickel in there because molybdenum and nickel, their key process in terms of physiology is in nitrogen metabolism. So when you have molybdenum or nickel deficiencies, sometimes you get a general yellowing on the plant, which appears as nitrogen, and may not necessarily see a molybdenum or nickel deficiency on a new growth of a yellowing. So sometimes these symptoms can be a little bit difficult to pull out of a plant just by visually inspecting it. And this is when tissue analysis will need to be done for these plants to confirm that your deficiency symptoms are exactly that, because a deficiency symptom has to be confirmed with tissue analysis. If you see this being repeated time and time again, then you definitely need to be looking at tissue analysis. They are costly, but sometimes they're essential in understanding what's going on in the plant. Now the macronutrients, as I said, are present in dry tissue concentrations as percentage. The sulfur and calcium, even though they are macronutrients, they are relatively immobile in the plant, so their deficiency symptoms are going to show up on the new growth or the newer growth. Calcium deficiency is really dependent on transpiration and water availability in the plant, so you're going to see very odd deficiency symptoms with that. On this slide, I have a few examples of mobile nutrients and their deficiency symptoms. These slides were used from the American Phytopathological Society through copyright permission, and these give examples of different mobile nutrients. The one on the upper left is a nitrogen deficiency symptom in citrus. Now the stem on the right-hand side of that photo is a citrus tree that has adequate nitrogen. You can see all the leaves from the oldest to the newest are green. But if you look on the left, the oldest leaves on that stem are turning yellow, and they're starting to senesce and they fall off, and that's why you have no leaves below there. And so that's a nitrogen deficiency symptom, very classic. It's a general yellowing of the leaf. And this happens very commonly in many plants. The symptoms are very similar, a general yellowing of the leaf, and then they'll drop off usually, or they'll senesce off. On the right-hand side is a photograph of chrysanthemum, and this is an example of a phosphorus deficiency in plants. These deficiency symptoms you'll see sometimes, in many cases with tomatoes, you'll see a purpling or a reddening of the stems with phosphorus deficiency symptoms. You see pigmentation. You also see stunting and stunting of the roots. Sometimes phosphorus deficiency is just because roots haven't developed properly. And this is often the case in the field where there's cold soils and the roots are just not growing adequately. And so even though there's phosphorus present in the soil, it can't be taken up by the plant because the root system hasn't developed yet. But you see a general stunting of the plant here, as well as a slight reddening, although that's harder to see. On the lower left is a potassium deficiency symptom in blueberry. This is a very classic symptom of potassium deficiency. This leaf on the left is the one that's deficient, and the leaves on the right are the ones that are not deficient. And you can see that the leaves on the left, you see a reddening and a senescence on the outer leaf margins. And what's interesting with potassium deficiencies is it's not like nitrogen where you get a general yellowing. You get a necrosis in orange, a collapse of the tissue. And the reason you get this is because potassium is very important in stomatal regulation, stomates opening and closing. So when there's inadequate potassium in the leaf, the plant can't regulate water loss. So you basically get what looks like a drought stress on the oldest leaves. You get a senescence on the outer part of the leaf tissue. First orange or reddening orange, and then these symptoms will coalesce and you'll get total collapse of the tissue. So sometimes potassium deficiency can be confused with drought stress or even salt stress, where salts accumulate on the outer parts of the leaves. So this is again another example where at times you may have to rely on lab analysis, not just visual observation, to determine what the problem is. On the last photo in the lower right-hand corner, this is a crop of chrysanthemums grown for cut flower. And if you look in the lower leaves, you're going to see the lower leaves are yellowing on this plant, but the veins are still green. So in the case of magnesium deficiency, unlike nitrogen or potassium, you get a yellowing in the intervenal areas, but the veins will stay green in the plant. Now what's interesting with magnesium deficiency in this example is the plants are flowering already. They're fully mature. They're ready to be harvested. So yes, there's a magnesium deficiency here, but it serves no purpose for the grower to go in and apply Epsom salts or something else to rectify this, because he's not she's not harvesting the leaves. They're harvesting the cut flower stems. So in this case, we don't have to worry about it, and we can leave it go. So again, if you were in a different crop system where you had the magnesium deficiency symptom and you were growing the crop out repeatedly or maintaining it for another year, you would have to go in and apply some sort of magnesium to fix this problem. Unfortunately, for things like the magnesium deficiency in the lower right-hand corner, or the nitrogen deficiency in the upper right, and some of the other nutrients, if the leaf tissue is too far gone and has remobilized those nutrients excessively from those leaves are not going to recover. And you're just going to stop the symptoms from progressing onto newer growth. So depending on how far gone or how severe the symptoms are, some leaves will not recover adequately when you go and apply the treatment, even if you do a foliar application. Because in many cases, foliar applications, depending on the leaf type, will not penetrate the leaf in adequate amounts for the deficiency symptom to be corrected. Citrus is a very waxy leaf. Avocados have very waxy leaves, and it's really hard to get nutrients through a foliar application into the tissue. Now the immobile nutrients, again, calcium and sulfur, even though they're one of the macronutrients, they're present in a percentage level in the leaves. They are immobile in the plant, so their symptoms will always show up in the newest growth. With calcium, you often see it in fruit disorders, with bitter pit in apple and blossom end rot in tomato. Sulfur historically was not run across very often with deficiencies because of the air pollution, and now we have cleaner air, and symptoms of sulfur deficiency are coming up more, especially in rural areas of agriculture. The other micronutrients are all immobile, and so their deficiency symptoms will show up in the newest growth: iron, manganese, copper, boron, and zinc. And because these nutrients are required in such small quantities, oftentimes you'll rectify the situation. You may not necessarily know what exact nutrient, if it's just one nutrient or several of the micronutrients that are deficient, but you can buy chelate packages. These are micronutrients that are chelated. They are actually in a form that's soluble, and so you apply these chelates and they contain many of the micronutrients on there. It's important because if you apply too much, you end up with micronutrient toxicities and can burn the plant. Now I'm going to give an example of some of the micronutrient deficiencies and how they appear in the plant. Again, these deficiency symptoms will be on the newest growth. And for example, on the left, calcium deficiency symptoms in citrus, you will see the oldest leaf is the dark green one on the left, and the newest leaves are the ones on the right. And calcium deficiency kind of looks like a little bit of a magnesium deficiency, but in this case, the deficiency symptoms are going to be on the new growth. A more classic example would be in tomato production or in fruit production. And what's interesting with calcium is that it's really reliant on the transpiration stream as well as the stage of development of the plant. So when a flower is pollinated in a tomato, for example, there's a lot of cell division going on. The cells are being laid down, and calcium is very important for cell wall integrity. And what happens is, if you have inadequate calcium in the soil, or your irrigation is such that you didn't have enough water on the tomato plant when the cells were being laid down in the new tomato, the cells are already laid down and you go and you water, it's too late. It's already past that physiological stage of development, and even though water is being reapplied and calcium is being taken up and taken into the plant, the cell structure, the cell walls have already been laid down, and so the tomato looks normal in the beginning. But as the tomato goes through a next phase of growth where the cells start enlarging, the cell wall becomes larger, but it's weaker, and so the cells collapse. And you'll see blossom end rot in tomato. In apple, it's called bitter pit, or yes, I think it's bitter pit. And you'll see pitting on the outside of the apple, and that's due to calcium deficiency during cell division stage of that fruit. Iron deficiency is very classic and it's very common. The reason it's common is because iron is very, very dependent on the solubility in the soil. And oftentimes, especially in the West, where we have very high pH soils or alkaline water, iron is very immobile in the soil. It's precipitated out, it's not solubilized. And so iron deficiency symptoms show up not because there's not iron in the soil, but because the pH keeps this iron precipitated out of solution. A way to work around this in the industry, especially in the container industry, is to use chelates. And chelates are like a hook and claw thing. They hook the iron to the molecule and keep it in solution so that it doesn't precipitate out. But iron deficiency symptoms will show up on the new growth. And again, it's a very fishnet appearance. You'll have the veins will be green, but the intervenal areas will be yellow. And so you'll see that. And if this gets worse, the tip will die. You'll get necrosis of the new leaves, and they'll be deformed. Another example of iron deficiency, a better one, is down here in the chrysanthemum on the lower slide. And you'll see that the iron deficiency, you see the veins are green, but the leaves become yellow, and the chlorophyll has broken down. A problem with this sometimes is they're also very sensitive to the light. And so if you have high light, sometimes you'll get necrosis as well of the tissue, and may make the symptoms a little bit different. And you'll see necrosis of the tissue. Now with iron and some of these micronutrients, when you apply the micronutrients to the soil or foliar, oftentimes you will get a recovery of that tissue and it will grow normally. But sometimes that isn't the case if the symptoms are too far gone. Another example of a micronutrient deficiency would be manganese, not magnesium. Manganese is a nutrient that occurs, deficiency symptoms will occur in the plants, and the new growth will come out with the leaves folded. This especially happens in monocots like palms, especially in Florida where the soils are cold in the wintertime, and there's not enough manganese going into that new growth. And as the season progresses in the spring, it's warm, those leaves will come out in an accordion-like fashion. They will be deformed and they will never recover. And the new growth comes out normally because the root systems are functioning and growing actively in the warm soils. As I mentioned earlier, the nutrient deficiency symptom photos were from the American Phytopathological Society. And if you would like to access these photos or other types of photos, they have a CD collection that's available that will go through all the plant nutrient disorders. They also have other collections of other nutrient and abiotic and biotic disorders in plants. And you can access this collection from the APS site, and will help you with some of these symptoms. Okay, I want to thank you for your time, and thank you.